very interesting talk on. It's on right now.
Oh, should we postpone? Okay.
I don't know. I just thought you'd be interested. It's phase relationships in phototaxis of microswimmers.
Okay. Maybe.
Maybe, yeah. Well, they're.
Can you get a video of it?
I don't know if they'll have them on later or not.
Okay. Well, send me the reference. I might just write the author.
Yeah. Just look up papers that these people have. I'll find you a few.
Yep. Okay. Oh, good
That's
morning.
great. Good morning. How is everyone? Any news this week or?
No, took care of grandchildren all weekend, but that's all.
Oh, okay.
Well, I babysat dogs and a cat.
Oh, wow, yeah.
Just what I wanted to do.
Yeah.
Oh, that's probably harder because they don't play video games all day.
Oh, there's a cat behind me, yeah. If it isn't one, it's another.
Yeah.
So, yeah,
This
thanks.
cat's very demanding. Meow! Okay, what do you want? Meow! It's like, okay.
Ooh.
So thanks to Dick for submitting that paper. Finally.
Yes, finally. Only 3 months. It was only 3 months late.
Yeah.
I'm surprised they still accepted it.
Yeah. Yeah. That's great. And Susan, you went to APS 2 weeks ago, I guess.
Yeah, it was good. It wasn't always on topic, but yeah, I'm planning on doing kind of a synopsis for you, just some of the highlights that I found.
All right.
So yeah, a lot about cell mechanics. They had 4 lecture sessions on cell mechanics, and so I was happy. At least no one was doing what I was doing, so that's actually a good thing.
Oh, yeah.
Okay. Oh, we had to do some heavy lifting of some lumber this weekend.
Oh, okay.
Natalie made an interesting observation. Why is it when you lift something heavy, does your heart speed up?
Yeah, because you
Is that
need.
an integrity question?
Yeah, it's a feedback loop.
It's a feedback loop. Of what sort? From what to what?
I don't know, muscle movement speeds up heart.
Yeah.
Okay. Okay, so it's not, you don't think it's part of Levin's tensegrity effects?
Well, indirectly, of course,
Of course.
since we're a tensegrity object.
By definition, okay.
Yes. There you go.
Okay. Okie doke.
Yeah.
I don't know. I'm just trying to model cells with it and trying to do it in a linear manner, no springs. I ran into someone who said, "Oh, you should try it in Morphle." And I said, "Well, maybe, because if I could do it in several different platforms, then I would know that it was producing the same thing at least." And he said, "Oh, well, give it to me. I'll see if it'll fit." So I just gave him my stuff, which is probably the wrong thing to do, giving someone all your research.
Yeah.
It's like, I don't care. Copy my research, please. And anyway, he took it, and I heard later that he was a very nice professor, and that I was really lucky that he had taken interest in this. So it's just like, yeah. Okay. So
Okay. Susan, I put the name of another program that if you're gonna look at different programs or platforms. I used this a few years ago.
okay.
And I think I remembered the right name. Push me, pull me.
All right. Well, this Morphle, they have developed it.
If you can't find it, let me know and I'll look it up.
Okay. I'll just copy it. That. And it's the 20th already. Yeah. Okay. What is that? Push me, pull me?
Yes,
Yeah,
I think
yeah.
that's the name, but I'm not absolutely sure.
Okay.
I think it was developed in Italy.
Really? Okay. Okay, well, what I was going to try to use was Morphle, but I'm not going to tell anybody that I really did use it until it works.
Yeah.
It doesn't work. I never tried it.
Okay.
Yeah.
Okay.
Yeah. So that's great. That sounds good. Look forward to your synopsis on that.
Yeah. Well, it's always interesting, even if it isn't on topic. It's they go in 10 different directions or more, and it's all about cells,
Great.
cell tissues. So,
Yeah.
oh, it was, some of it was fascinating.
Like what kinds of things were they doing? Just
Hmm. Well, it was all work on either tissue mechanics or things like microswimmers. What is that? Modeling, statistical mechanics. Let's see. You'd be better to see a list of their things.
yeah.
And they had some work on macromolecules and protein folding, of course. And the ones with the stars on them are the ones that I was interested in, and brain organoids. They had lectures on brain organoids.
Great.
Okay.
Yeah.
I put a paper in, Susan Bradley, I put a paper in the chat.
Yeah.
This includes the possibility that diatoms can actually swim.
Right. Yeah.
They have cilia?
No,
No.
entirely unique mechanism.
Okay.
Yeah. So this is isn't this the paper where you have like 20 different motors? I think you presented on it.
Oh God, yeah. Well, I went through the history of models
Yeah.
of how diatoms move, and it's really amusing.
Oh.
You know, like one guy saw paddles.
Yeah.
Yes, and diatom moved the way those old-fashioned boats moved, with lots of slaves having paddles.
Okay. Yeah.
So I've heard of microswimmers. It's they're these little what are they, like cellular systems. So they're living systems, but they can study swimming at very small scales. So they're actually getting it down to like almost like the nanoscale, or that boundary where you have different hydrodynamics.
Yeah, it's kind of weird. I mean, the idea is basically the ray fluid is touching water, and at low Reynolds numbers, the effect of the ray fluid on the water may lead to propulsion.
Yeah.
Okay. So it's very speculative, but it has to be, a proper test needs to be done at zero gravity.
Yeah.
Okay, because diatoms weigh more than water, so they'll sink in the water by themselves.
Right.
So some of them float at the surface, because they're also, the surface, parts of the surface are hydrophobic. And so they will actually float at the top. And they seem to move around even if they're floating at the top.
Yeah.
Okay? So the question of their ability to swim is open.
Yeah.
Oh, Sushmanth, if you want to read papers like this, send me your email.
Yeah.
Okay, I'll put. Okay, that's my email address in the chat.
Well, did the student from the conference get a hold of you yet?
Me or Dick?
No, I'm sorry, Dick.
Okay.
I think I gave him that reference because of the waves. Like he had simulated some waves. See, that's what I mean.
Oh.
Interesting. He'd simulated waves that went out and then came back, and he'd simulated some waves that went out and dissipated, and then some that came in and dissipated. Like he had a
Okay.
and it depended
what
on
was-
the wave.
What was the barrier? They reflected off a barrier, or they?
No, I think it just went off into the edge of his simulation. But I'll get you that reference. I've got to go through my notes here and pick out there's 5 or 6 lectures
Okay.
that
Yeah, in the differentiation waves, we had 2 categories that we never were able. Well, we don't know what's the difference between them. Some waves seem to stop when they reach a certain barrier, and other waves seem to go through the barrier.
Oh, okay.
Okay, and we don't know the difference between those 2.
Okay, well, he had some very good simulations. Now, I'll just have to do this for you. Anything I've starred here. I took like notes for the whole thing, and it was 5 days long. And the notes are like this. Most of it was scribbler. So
Yeah.
yeah, need to go through that. And I'm still trying to do my modeling. I've given up on truss models, and I'm doing a solid model now
Okay.
in console. Yeah, that's my progress. Except I was putting in my last cylinder, and I wanted it to do this and attach from here to here. And what did it do? It did this, or it did this.
Huh.
And it won't go at an angle no matter what I do. I can tell it which points to go to. I can tell it what angles to go to, and it won't. So I don't know. It's a goofy program. This is why I was thinking, oh, Morphle, what does that do? How is this going to be any better than what I've got? We'll see.
Well, yeah, I'm sorry you had to give up on the truss structures. I mean, that was kind of
Yeah.
interesting. Yeah.
Well, it was nice, and it worked. But I can't get a stress strain curve out of it, and I asked them about 5 times how to do that. So if they don't answer me later in the week, I'm going to send them another note saying, how do you do stress strain curves in truss structures? Again, and again, and again.
Until you stress the person.
Yeah.
I don't know.
You do a stress strain curve of the
I'm going to
person.
stress strain them. Yeah, we'll try it. I don't know. Why does this always happen to me?
It happens to all of us.
Okay. Yeah, I'd be done the project if I had these curves. I could just say, well, the stress strain curve did this,
Yeah.
and here it is, and I'm done with this project. They said I could be done with it.
Yeah.
I've written 500 words. I just need to write like 10 times that, and I'm pretty sure I can. But I need my graphs.
Yeah. That's good. Thanks for the update.
Okay.
Sushmanth, how are you?
Yeah, hi.
Hi.
I'm fine, Bradley. How are you?
All right.
Yeah, last week I was completely working on Devoland. Actually, I have kept those 2 PRs. Can I share my screen and show it to Dick?
Yeah.
Actually, he mentioned that update. Actually,
Yeah, go ahead.
Yeah. This is the. I think he remembered, like he asked an update like centroids with different color and membrane with a different color.
Yeah.
So I added this visualization method.
Good.
And this is how it works. And upon of it, I added this also, like comparison of 2 different frames. This is the original microscopy image, and this is like a segmented one. And it's working fine. I have kept the PR, and it is much. Mike give me some pretty good.
Okay. Can I
How
ask
to keep a good.
a stupid question?
Okay.
It seems that the centroids move.
Yeah, accordingly, when they are like dividing up, they are moving.
Yeah, as you go through the section, you're going through sections, right?
Yeah.
And the centroid seems to move. Okay, now can you identify the true centroid of a 3-dimensional cell?
But we don't have like position of true centroid, right? Like for this data set, this is like segmented one, we are getting it out. But we actually don't know truly like where the real cell was there.
Okay,
Yeah.
but you have multiple sections, and so for a given cell, you can get the volume.
Okay.
And since you have multiple sections, the multiple sections represent a 3-dimensional object if you take just one cell.
Okay.
Therefore, the 3-dimensional object has a centroid.
Yeah.
And that centroid will be different from the moving centroids that you see when you go through the sections.
Yeah.
Okay, can you identify the 3-dimensional centroid?
Okay, I will try to find it out and I will work on it. I just went through this like last week, and I have worked on my instance segmentation model also, Bradley. I want to show you the results.
Because if you looked at them from the side, they would look like a 3-dimensional object with a that consists of a stack of closed lines.
Yeah.
And therefore, that object, if it can be called one object, has a centroid.
Okay. I have worked on my instance segmentation model. Like I was trying to build an instance segmentation model which can segment it. These are like results of it. I have plotted them on image 3D viewer, which you have mentioned last week. This is how are the results, except this cell, all other cells are segmented properly. You can see them. This is segmented image. This is like real image.
And if you look at the image, if you look at the image on the right, it seems to have contour lines.
Yeah.
In the individual cells. Are those contour lines correspond to the outlines in the sections?
I didn't get you. Like, can you repeat?
Okay. Look at the yellow one on the left. Okay, it's got a square on it. Does that square correspond to the shape of that section of that cell?
Yeah, this is the shape of that section. I mean, that's estimated. Actually, I was training a model to segment it properly. I'm creating a ground truth and I'm training the model. After that, when I trained it, I got these results. And
therefore,
and I'm trying to I'm trying to calculate the volume also for this each cell by using like first calculating the area of it.
So you already have you already have the data then stacked for each cell. So if you get the centroid of the volume, that would do it.
Yeah, that would give it. We can get centroids of each cell. That would be the nucleus, and volume also we can calculate. How means like we will take the OpenCV library. We'll calculate the area of pixels of each cell, which is it is occupying. After that, we will multiply with multiple frames. Then we will get the volume of each cell.
Okay, what I'm suggesting is that the 3-dimensional centroid is the one that one wants to track.
Yeah.
Not the 2-dimensional centroids.
these are three-dimensional centroids only. We can extract it. See, you can X, Y, Z. So I mean these.
Yeah, but then you could track the 3D centroids in time.
Yeah.
How they interact.
And how I think I have the GIF file also. Maybe I can show you that.
Okay.
Which it is like working very fine. Yeah, this is a GIF file. Sorry, it's not playing. I think I have in my docs. Okay. Just a minute.
Okay.
Yeah, this is the image. Can you see it? I hope.
Oh yeah, it's moving.
Clearly.
Right. Well, yeah, it's a little small, but you can increase the magnification of the file. Just
yeah. 5 minutes. If I crop it, maybe I can.
Okay, I have another question about these. It looks like they're cells that are floating in space.
Yeah.
Okay.
Yeah.
Is there any way to indicate whether two cells are actually sharing in the sense that they have a membrane in common between them?
Maybe we can do it. Like, I don't have clear idea, but I think we can do it. Which is easy only. Like, we need to membrane, right?
Yeah, each cell has a membrane, but if cells are touching, the membranes will be so close they'll almost coincide.
Actually just work on like extracting the centroids and volume of each cell. So I'll give it into a look of this membrane.
Yeah,
so I'll try to.
I guess I'm asking is, what I'm asking for is, can you indicate if any two cells are touching? If they're touching one another.
I think, yeah.
Because this right now looks like sort of a random. You know what it looks like? Have you ever played bingo?
Oh yeah.
Yeah.
Okay. You know how the machine throws the balls up in the air so they're randomly distributed? It looks like a bingo game.
Okay.
Moving around,
And,
yeah.
Okay.
Okay, I will try.
Yeah, the other information that might be useful is if there are a way to label cells so that you knew which cells were daughter cells of previous cells.
Yeah. Actually, this color may indicate that one, like from which cell it is coming. I will try to create it like which is parent cell, and like see this pink and pink color. Those are. I'm trying to create like that. After that, I want to create it as a mother cell and daughter cell. I will try to create it. I'm just working on it actually. To get these results only, it took me so much time. I'm pushing this code.
Let me push that to the limit. The question I'm asking, in a sense, is if you looked at the lineage tree of the cells, in 3 dimensions, the lineage tree would be highly folded in 3 dimensions. Can you draw the lineage tree on top of the cells?
Okay. I got it. Maybe I will try it. Like I clearly don't know. Like I know about lineage tree, but on top of it, cell, I need to check it out. Like, can I work it on?
I mean, the lineage tree changing in time, for example, is then a 4-dimensional structure, which is hard to visualize. But that may be what the embryos are using. Okay, so in a sense, I'm trying to ask questions like, is there a relationship between the lineage tree in 3 dimensions over time and the construction of the embryo? It sort of becomes sort of a global question. Because one thing we're really lacking in embryology is an understanding of how the embryo controls the whole thing. And by looking at the lineage tree over time in 3 dimensions, one might get some ideas.
Okay, I got it. I understood the question. I need to see I can do it or not. Like I need to see implementation. I think I have read the research paper on lineage tree, but I don't know the correct implementation of it. I will
Yeah.
try it on.
Yeah, it
I
also
will
gets
see.
complicated because you can't see the whole lineage tree because the older parts have disappeared as time
Yeah.
goes. Okay.
Sorry. One minute. Actually, Bradley, with this we can extract topological data analysis also with this shift file. Like
Oh yeah.
I mean, with cells we can we are segmenting cells. We can get like how many dead cells are there, how many alive cells are there, etc. etc.
Yeah.
The toolkit which you have shared actually is like pretty good, and we can work on it. This one, I'm working on it. I'm trying to get the data analysis of it. First, I was creating a pipeline of this instance segmentation model. After that, whatever data is extracted and giving it to the data analysis, I'm trying to build it out. I have kept the repo privately for some reasons. After like proposal submission, I will keep it open and you can review it. And I can show you, like I just wrote.
Yeah.
I haven't pushed the whole.
Yeah, there's the. Oh, I think you froze up a little bit. Sushmanth, are you there?
Whole code actually. And another update I want to give is that Hugging Face is actually stopping after like 4 days or 5 days. I want to clear out that problem. I am just creating an alarm system. Like whenever it stops, like it will restart the space and it will work it on. Like you can use it. And this DevoWorm GitHub.io also. Can I update these links actually? We have the model, but I think we need to update this. Here, my what we call that heruko app is there actually. I was thinking to create another page by clicking this thing. We'll get a page like mention about Devoland and models of it, and membrane segmenter model, nucleus segmenter model, lineage population model. These 3 options we will get. After clicking that, it will take them to the Hugging Face page of these tools. Can I make that update to it?
Yes, please do. Yeah, that would be great.
Okay.
Yeah.
Two more, or a couple more comments.
Okay.
First of all, in terms of display, are you familiar with the optical illusion that if you make a projection on your computer screen of a 3-dimensional object, but the object is rotating, it then looks 3-dimensional?
Yeah, I know about this thing actually.
You know about that? Okay, so one thing you might do is rotate the embryo as you're displaying it.
Okay, actually I think I can rotate this one only. Maybe by next meeting I can show you. Whatever this image is there, right? This toolbox, we can rotate it actually in.
Oh yeah, I'm just I'm sure you can do it. I'm just saying if you rotate it during display, it may appear more 3-dimensional. Okay, the other question is, do you understand the difference between a lineage tree and a differentiation tree?
I know about lineage tree, but I don't know about differentiation tree.
Okay, the differentiation tree, all of the cells that are of the same type are labeled.
Okay.
So sometimes you get a cell division so that both cells are of the same type. Sometimes there are different types. This is true in nematodes. There are about 50, I think in an adult nematode, there are about 50 pairs of cells where the cells are identical. Okay? So a differentiation tree lumps the cells together that are of the same kind. Okay?
You have any research paper regarding it, like a differentiation tree?
I have a small paper I wrote years ago on that. It shows it schematically. basically it shows how differentiation tree includes the lineage tree but lumps it together.
Okay, okay, got it. This is what you're asking, right? Like when we rotate this frame, it should give like name on the top of this cell, like whether it was like parent cell or a daughter cell. That's what update you're
Well,
asking,
that's
right?
sort of it, but basically, if you have two purple cells, then they are the same kind.
Okay, yeah. That's what I
Okay.
want to mention. But
Yeah, that
I was just
that would
want
be an example.
yeah.
So the differentiation. So actually, what we're after is, does the differentiation tree change over time in a way that explains the three-dimensional structure, or I should say four-dimensional structure of the embryo?
Okay.
Okay, these are very tough questions because you're trying to get 4 dimensions onto a flat screen.
Yeah.
Okay, it really twists your brain. Yeah, it twists your brain.
I think by next meeting I will label it. I will label these images like on a 3D viewer, like this is a parent cell. When this diff file is running right here, okay, I will let me get this is I will just create a 3D kind of thing, which when we rotate it, we can see like which is parent cell, which is daughter cell. So
Okay.
I can get a lineage tree of it. I'll try to make it. I just have my midterm exams actually, so I was really going through them.
Yeah.
And I will try to keep a PR. Like I will try to work on it by this week or next week. Next week also got like pretty busy weekend. I'm going to New Delhi or work on some project actually. Visiting Indian villages and finding out their problems and trying to make a research paper out of it, like by solving them. This is a C20. Anyone know about G20? Like Government of India, some initiative is there going on. As a part of it, our college is like going to villages, Indian villages. We are visiting them. We are finding the local problems there, and we are trying to implement it. They are like, how can we solve, as an engineer, how can we solve a problem?
Yeah.
And after that, after a good project, we will try to make a research paper of it. Next 2 weeks, I was pretty busy with that. I will try to work it on. After that, I'm completely free.
Okay.
And the proposal also, do I need any changes? I tried to add the data analysis part also, topological data analysis. Mayuk reviewed actually 2 days back. He reviewed it again. This is like third phase, I think.
Okay.
And he liked the idea. He told like segmentation results are good, but he just told me to calculate volume properly of each cell.
Okay.
Actually, DevoGraph really needed it. I mean, it's the volume of each cell. So I'm working on it actually. He gave me this idea of calculating area of each cell. After that, keep them in a frames, video frames. You will get the volume of each cell. That's what idea he told. I'm trying to work on it. Maybe I can complete it. And I think today we should start submitting proposals. Like when should I submit? You will review it again, Bradly, or should I submit the proposal? These tools need to be updated actually. I will try to update it. I don't have knowledge of web, but I will try to do it by this weekend.
Yeah.
Let you know.
Yeah, it's been out of date for a while. We've kept working on it, and we built that a while back, but it always needs to be updated. So yeah, please do. This is on GitHub.io, so this is a different repo. This isn't the DevoLearn. This is the DevoWorm GitHub, and it's like the GitHub.io repository there. So that's where that's hosted.
I will work on it. I will just see and I will update those links, and that's what my updates are.
Okay.
And if you need any changes, like you have any suggestions, then.
Oh yeah. I mean, we can just update the links for now, and then we can go through it later. Because but it's probably though. If we were to do an update, it would probably be at the end of the summer, once we get more work done. Yeah.
Okay, I will just update it by tomorrow. Like I can do it, but these exams are Okay, I
Oh
will
yeah,
just
yeah, no
update it
problem.
and I'll
Yeah,
let you know.
do it when you can. Actually, it's
Okay.
fine. It's been outdated for this long. It's
Okay. Okay.
but yeah, that's
Uh,
great. Well, thank you for that update. That's a lot of work. It's great work.
okay.
And so I'm gonna share my screen. So if you're applying to GSoC, we are at the first day of being able to submit proposals. So if you want to submit your proposal, this will be the first day that it's open to submit. But of course, if you're submitting a proposal, you'll want to, talk to me about, kind of the structure of the proposal and the schedule. Because our projects are rather open-ended. Sushmanth has been talking with me about his proposal, and he's been doing contributions, prior to GSoC, which isn't required, but it does help clarify your proposal. And then the project is a half-time project, so it's 20 hours a week for 12 weeks, and so keep that in mind when you're writing your schedule out. So this is the DevoLearn repository here. This is the organization that we're working with for this GSoC. We have the DevoLearn repository, which has the EvilLearn software in it. This is the software. This is the first software package that we developed in this DevoLearn repository. So this was developed in 2019, 2020, 2021. So in that period, we built this pre-trained model. It's built on, it's a deep learning model. It's built on Python. You have some example notebooks. And this is where we're able to get some capabilities for segmenting cells. Although, we've been working on refining how that's handled. As Sushmanth has pointed out, this is semantic segmentation, which is actually useful for, finding things have categories like tissues or specific, cell types or cell groups. But you also want to have instant segmentation where you have individual cells that may be unlabeled or just kind of like in a early stage embryo where there's no tissue differentiation. So we have, training data for this. We have, the idea is to take a set of images that you can get on the web or, from an experiment that you're running. You can plug it into this tool and then you can get segmented, ideally 3-dimensionally, but, 2-dimension. Usually images are acquired in 2-dimensional slices from the dorsal to the ventral side of the embryo. And you take those slices and you can analyze them, getting segmented cells, getting volumes, getting centroids, and then having those data outputted as a table or in a tabular format you can then use to build like a...
Bradly,
Yeah.
I had a comment here. That picture that's rotating there was a single cell.
Yeah, these balls here are single cells. So these are, I think, centroids of those cells just kind of put in this 3-dimensional space.
Each one is a cell.
Yeah, it's a centroid of a cell. So it's just a representation of the distance between the cells. So you can see it's an embryo here.
Okay, now you're familiar with confocal microscopy?
Yes, yes.
Okay, in confocal microscopy, you're trying to get a sharp image of a given plane. Okay, however, because you're looking down the axis of the microscope, you get what's called Z or Z blurring. Okay?
Yeah.
And this gives you an asymmetric point spread function for the image.
Great.
Okay. Now there are a few papers. I think I wrote the first one on how to deconvolute this point spread function to make a sharper image.
Okay. Yeah.
So you might want to look into deconvolution of point spread function of the confocal microscopy image.
Yeah, that's
I'm
actually
sure
interesting.
there are probably some papers on that by now.
Okay. Yeah, I'll take a look at that. So that's where you have these slices and you have them at different focal planes.
Yes,
and you're looking
exactly,
down.
exactly.
Yeah,
It's trying to make your focal planes thinner
right.
by deconvoluting that point spread function.
Right. So that's so we have a lot of stuff in place, and of course it can always be improved upon. That's what we're doing here with some of the pull requests that have been issued here. We have let's see, a couple that are closed. I think Sushmanth has been working with Mayuk. Mayuk Dab was the person who first put this organization repository together. Then his brother Maynak worked on it in 21, and then in 22 we worked on this a bit with respect to the DevoGraph project, and now we're working on this year. So Sushmanth has been working with Mayuk on this. Mayuk is the maintainer of this repo, so you might want to be, if you're interested in contributing, you want to be in communication with him. But we have a lot of. I don't think we have any. We have a couple open pull requests, but I think those are just kind of leftovers from last year. So, but yeah, so we have this DevoLearn, and then we have, of course, this is released, in different editions. So we have, I think the release right now is 0.2.0 or 0.3.0. 2 releases. So we have, we're at 0.3.0 right now, and this was a while back. We were going to do an update last year, but we didn't get to it. So we'll probably do a release at the end of this year, at the end of this GSoC, and hopefully it'll have DevoGraph in it as well, but also the updated capabilities for DevoLearn. The release just means that we have a formal release people can download that isn't changing all the time due to pull requests. We have a PyP site for this, so we have it, in a place where we can collect statistics on it. Then we have the DevoGraph, which is, of course, this project or this repository. This is from last year's GSoC, and Sushmanth has been working on this. Gautam has been working on this. A couple other people are in the process of forking this. So we have 6 forks right now. So we're working on these different aspects of graph neural networks and building a pipeline for this. But we have to sort of make sure that DevoLearn, the original package I just showed, is working well for all different types of applications we want to use. That's kind of like the core of this whole thing. So DevoGraph is basically taking those data that we've collected in DevoLearn and turning it into some sort of graph embedding so that we can do things like build trees and build network topologies and things like that. So if you're interested, again, we have no open pull requests. We had a couple Sushmanth, I think, and Gautam have pushed some things and they've been accepted. So if you're interested, just file a pull request, define it, describe what you're doing, and then we'll review it and hopefully accept it. So that's great. Also wanted to bring people's attention to the Slack. We have not only, if you join the OpenWorm Slack to join the discussion about GSoC, you have the DevoWorm channel, which is, of course, the main channel. We have a lot of people coming in, I see. I think in the last couple of weeks, we've had about 10 people here join. Again, you're welcome to join our meetings. I'll post an announcement for the meeting about 10 minutes before. And, you can come in and please discuss things in the DevoWorm channel. But we also have a DevoLearn DevoGraph channel. So if you're working on, say, the repository, or you're interested in some aspect of the project, of the GSoC project, you'll want to do that discussion in this channel. And so we have a lot of different things that we post. I posted some readings on, these are from last year, on topological data analysis. So, there's a paper on C. elegans locomotion, Python tutorial, this shape of things to come, which is actually a nice review article. expressivity of persistent homology. So in graph neural networks, there's this idea of expressivity, or what can the algorithm do, or what can it represent? And that's actually a pretty popular topic in graph neural networks. You have this idea of expressivity, and if you write an algorithm or if you build a representation, how expressive is it? What can it capture? And that's a topic we can talk about, but it's something that is very important, because if you're building a model, you want it to be able to express what you actually are interested in. So there are a lot of things here, and then we have I have some more readings here. Graph to graph neural networks. This is a review article from on someone's Medium, and then Sushmanth posted some things on pull request. Jiahong Li he worked on the graph neural networks last year, so he's been in and out of the Slack recently, but he posted this last week. He posted a couple of I think he posted his proposal from last year. So if you're interested in reading a proposal that got accepted, you can read his proposal, and that has the proper structure for a GSoC proposal. So that's the place you want to go for that. He also has a couple readings here on graph neural network for cell tracking and microscopy videos, topological graph neural networks. This is merging topological data analysis and graph neural networks. And then the DevoGraph library, which is another tool you might be interested in if you need to, because graph neural networks have a very high barrier to entry. It's just you have to learn a lot before you can really do a lot of work on it. So that's why we've been struggling with this for last year and then this year. It's not really easy. It's hard. It's a new area. It's kind of like a Wild West. There are a lot of different types of approaches people are using, and so whatever you can come up with is probably it's probably a good contribution. I'm not saying that you can just do anything you want. It's just there aren't a lot of solved problems in this area. And then, of course, the shape of things to come, which is this topological data analysis review specific to biology. And that's something you'll want to read if you're interested in this project. So ultimately, we want to apply these graph neural networks to biological systems, to biological data sets. And so we've done a lot of work with networks, with trees, and this is going to fit into that work. And if you're interested in that work, we have our website, DevoWorm@weebly.com, and that has a publications page. You can check out the publications there. Okay, so I'm going to talk actually about, there's a new paper on graph neural networks from DeepMind, and this has just come out in the last month. This one's called Graph Schemas as Abstractions for Transfer Learning, Inference, and Planning. And this is a set of authors from DeepMind. Dilip George is a pretty famous person in this area. He's done a lot of really interesting stuff with AI and machine learning and deep learning. So if we look at the abstract here, so this paper is, we propose schemas as a model for abstractions that can be used for rapid transfer learning, inference, and planning. Common structured representations of concepts and behaviors, or what they call schemas, have been proposed as a powerful way to encode abstractions. So basically, they have these abstractions, which are these, I guess they're models for different things you want to do. They're, abstractions mean you take a process and you extract the essential aspects of it. So it's kind of like model building. You take a process, you observe it, you say, these are the most important things we want to take and put into our model, and that's your abstraction. So you can have different levels of abstraction. You can have a high level abstraction or a low level abstraction. If we take the embryo, one of the things we're doing in DevoLearn is we're taking the centroids, we're taking the volume of the cells, and we're using that as a proxy for embryogenesis. We're not talking about genes, we're not talking about physics necessarily, but that's because that's the model we chose, that's the abstraction we chose. And so if we wanted to, we could build an abstraction with many, many different details, but we don't do that. And then, certain abstractions are good for certain things. What they're saying is they want to build a schema as a model for abstractions. And so you have this, so you can encode abstractions in different ways. You have these schemas that they propose that can be used to use as a model for that. Latent graph learning is emerging as a new computational model of the hippocampus to explain map learning and transitive inference. So in this case, the hippocampus is a part of the brain. It's the part of the medial temporal lobe, and it's basically the center of memory and spatial learning. So if you're navigating a space, for example, your hippocampus is working, and there's specific cells that encode different types of maps, like place maps and grid maps and things like that of the space that you're interacting with. And so those are used as landmarks or as a framework for you understanding that space. And so you can use latent graph learning to model the hippocampus and the ways in which it does inference and learning. We build on this work to show that learned latent graphs in these models have a slot structure, or what they call schemas, that allow for quick knowledge transfer across environments. In a new environment, an agent can rapidly learn new bindings between the sensory stream to multiple latent schemas and select the best fitting one to guide behavior. To evaluate these graph schemas, we use two previously published challenging tasks, the memory and planning game and the one-shot street learn, that are designed to test rapid task solving in novel environments. Graph schemas can be learned in far fewer episodes than previous baselines and can model and plan in a few steps in novel variations of these tasks. So now what they're doing is they're taking these graph schemas that they're building of the hippocampus and they're using it in an AI system. So they're actually able to build a schema of what's going on in the brain. They have a model of it. They have this abstraction of the important parts of what the hippocampus is doing. And then they build the schema, which is a graph of that. So they're able to build a relational database, essentially. And that will allow you to build a better model of an AI for, navigation than, say, like a typical standard neural network. And so this is the reason why we do a lot of this as well. We do graph neural networks because we can build these graph representations. So sometimes things in the world have a graphic representation. So, there are a lot of things in the world that have sets of relations between them. They could be maps. They could be, structural networks. They could be just, networks of interactions. And the idea is we want to build these graphs that will tell us basically the essential aspects of those relationships. And then we can build, we can use that for AIs. We can use that for other types of data analysis. So it's really useful to do. And that's kind of what people are doing with graph neural networks. They're going beyond sort of the typical layered network that's, has weights that, you have to figure out what the weights are. The idea is that you have some structure in there and you try to recover the graph structure, the real graph structure, but along the way you still have this structured representation. We further demonstrate learning, matching, and reusing graph schemas in navigation tasks in more challenging environments with aliased observations and size variations, and then show how different schemas can be composed to model larger 2 and 3D environments. So this is just kind of their work on this. This is quite a long paper, but it kind of talks about how they've observed what's going on in the hippocampus, that you have these different, cognitive models that people have built. And then, using that as a basis for kind of extracting an abstraction of that so that we can build these graph schemas. And then these graph schemas will allow us to, model this as an artificial intelligence. So, there are different ways that you can do this. We build these schemas using clone-structured cognitive graphs, a computational model of cognitive graphs using high-performance computing. In particular, this work showed that latent high-order graphs can be learned in highly alias settings using a smooth probabilistic parameterization of the graph learning problem. So this is, there are a lot of problems, like this comes from graph theory. And there are a lot of problems in graph theory that involve finding the optimal graph for a given set of relationships. So that's one of the things you're trying to do. And, they've built these kind of graph, these graph schemas or these representations that can generate different graphical relationships. And then they try to find the best one to fit the problem. So, but we still have that graph relationship. So we're not working blindly. We're working from like a set of relationships. We just need to find the optimal set of relationships. And then, this is work that people have done in cognitive modeling. So, if we apply this to development, there are going to be different types of tools we may need to draw from. Unfortunately, there isn't a lot of work at all on this in the brain. In neuroscience, there's a field called computational neuroscience, and they worked on some of these problems, so they now have some idea of like maybe what the graph structure should be. But in development, we really don't. So that's going to be something we're going to have to kind of figure out on our own. We've done some work on like graph embryo networks, but those aren't going to help. I mean, they may help a little bit, but they're certainly not going to. They're not as developed as say some of the work that's gone on in computational neuroscience. So there are all sorts of problems you can apply to this. For solving simplified relational tasks, there are different benchmarks and other types of work that you can draw from. So this is a diagram here of using graph schemas in the CSCG model. So this is your schema matching with binding. You have these experiences and you have these schemas that extract from it. So what you do is you observe the hippocampus. There are these different experiences. There's this network of neurons that get activated. And then you have these schemas you can extract, which tell you something about the function. Now in development, we have kind of like, we have real lineage trees that we can draw from, or we have real differentiation trees that tell us something about maybe like the experience of a certain embryo. How does development unfold? Which cells divide and turn into what tissues, or what cells divide and proliferate at certain times? So we have these analogs in development. We can extract schemas from these networks, which means we're just taking like, maybe different snapshots of the data and saying, maybe this is something that will lead to, a good structure to work with for what we want to do, passing them on to, I don't know, some application we'll build in the future. Maybe they'll perform really well as an algorithm, maybe they won't. Then you have these cloned hidden Markov models. So you have, observations and clones. So you have these latent states. So you have these sets of observations, and then you have these latent states where they're not observed, but they're, hidden Markov model are these transitions, these different states that are hidden from view. You're not observing them, but they have to exist because the system has a certain structure that suggests that they're there. So you can infer latent states that you may not observe. If you watch an animal behave in space and navigate, certain parts of the hippocampus will be active. Maybe if they behaved in a different way in another environment, other parts of the hippocampus would be active, but you can't observe that, but they're what we'll call latent states. And so we can build a model of those latent states. And then we can build a model of continuous activation. This is like a continuous behavior where the organism is planning to some goal state. And so, this is actually relevant to embryos because, you have a process that unfolds to, say, the adult phenotype. So the question is, can we build a model that, goes from like an egg, one cell, to an adult phenotype? And then what paths are possible? Are all paths equally possible? Or we know from development that that's really not true, that there are certain paths that are viable and certain paths that are inviolable. So like in C. elegans, you have, the lineage tree always unfolds the same way, except when you have a defined mutant, and there are differences in that lineage tree. In something like a mouse, where development is not as deterministic, you can have different types of lineage tree topologies that can unfold in different ways, and they just use cells in different ways. So this is something, if we want to go beyond C. elegans, that becomes useful.
Brad,
Yeah.
I'd like to say something.
Yeah.
The notion of a goal in embryology is very difficult.
Yeah.
Because the assumption overall is that an embryo has no idea what it's doing.
Right.
Okay, it's developing into an adult, but it doesn't have the adult as the goal. Now, I can't find the quote, but I remember somebody saying, if you have an aberrant embryo, it goes towards the goal of being an aberrant adult, no matter what you do. Okay, in other words, it doesn't have the normal adult as its goal.
Right.
Or maybe there is no goal.
Yeah.
Okay. Now the in-between compromise on this is the Janus-based approach, where the differentiation tree consists of a set of small goals. The small goals, for example, are how far does the differentiation wave go, and what stops it. Okay,
Right.
so it stops at a given point, and that's sort of like a relay race.
Right.
If you imagine a relay race on an open field, which could go in all different directions, do you have any control over what happens?
Right.
Okay. Okay, there is no goal point. In other words, there's no point where you cross the finish line.
Right.
So the control of the development towards an apparent goal is one of the outstanding questions in embryology.
Right. Yeah, that's good. And yeah, I mean, when we compare it to the behavior in the brain, there are certain things you have to keep in mind that are different. But yeah.
Yeah, I had one young lady, for example, do a computer simulation of diatom morphogenesis, where she used the goal as the controlling thing, so the loops were all backwards. Okay.
Yeah.
So you have to, and she got the right result because the goal was programmed in.
Yeah.
Okay, so the question of goals in embryology, I think, needs a real critical discussion as to whether or not they actually work. Now, one example, have you read Megara Maruyama's papers?
No.
No, okay. I'll try to send them to you.
Yeah.
He was a sociologist who wrote a very interesting paper. It was called The Second Cybernetics.
Oh, okay. I think I may be familiar with it.
You may be familiar. Okay,
Yeah.
and it basically was looked at what you might call controlled explosions.
Oh, okay.
Okay, in various fields, and it was quite an inspiring paper. It keeps the problem is when you have something where the slightest deviation can cause a huge error because, for instance, you're exponentially increasing the number of cells. How on earth do you keep the adult goal in mind without having a mind? Okay.
Yeah, it sounds like chaos theory, actually.
Yeah, this preceded chaos theory.
Yeah, yeah, it did.
Yeah, his work was, I think, in the 60s.
Okay. Yeah.
Okay. At any rate, so that's, I think that's what bothers me about this paper. Yes, you can have a goal in a mind, but can you have a goal in an embryo?
Yeah.
Which has no mind.
Yeah. So I mean, yeah, they're not talking about embryos in the paper, but
No,
yeah,
of course they're not. Of course they're
Yeah.
not. I understand it's applicable here, but you got to be very careful about applying this kind of modeling to an embryo.
Yeah.
Okay.
Yeah.
Otherwise, you say, here's the adult embryo, become the adult.
Yeah. So yeah, I mean, this is basically, you know, the key here though is that you're trying to extract these graph structures that will tell you something about, you know, different contexts, different systems, and you know it'll have, as long as it has a sort of graphical structure to it, which means it has a set of associations, or it has this, you know, maybe this unfolding of things in time, that, you know, it can be used as a way to characterize the system. And then we have that, you know, that graph neural network, which would allow us to process the, you know, things in that,
Yeah,
within that
but do
framework.
they discuss deviations?
Don't think so. They just
Yeah,
kind of talk
in
about.
other words, the system sort of just unfolds. But if every step involves some randomness or some small error, how do you control those errors so that the end point is reached?
Yeah, I think they're just optimizing the networks in the things that they're
Well,
doing.
yeah, that's fine. That's fine
Yeah.
for mental things, but I don't see an embryo having an optimization brain.
Right. Yeah. Yeah. It's not. Yeah, so yeah, I mean, a lot of the you know, a lot of the machine learning literature and deep learning, it's, you know, it's all about like minimizing energy or optimizing the result. And of course, in algorithms, you know, that's kind of what you want to do. But at the same time, that can, you know, if you have like a generative algorithm or a generative process that's not really an algorithm, it's not algorithmic necessarily, then optimization isn't really what you want or going to get. Yeah.
And at each step, you've got variability.
Right.
And how do you keep that variability from going haywire?
Yeah. Yeah. So, you know, there are different things, like in that context, you know, there are different ways that people have approached this in the artificial life community. For example, they've used open-ended models and open-ended algorithms to sort of approach this, because if you're modeling like an evolutionary system, you know, evolution is producing outcomes that are, you know, variable. So you're not optimizing things.
Yeah,
You're producing.
you're taking advantage of variation.
Right.
But for an individual embryo, you don't want that variation,
Right.
even though you're stuck with it.
Yeah.
Okay. So these global questions: how does an embryo handle variations and still produce an adult?
Right. Right.
Okay, these are still open questions.
I'm just taking some notes.
Okay.
All right. Yeah. So that's that's the paper, and they go through a bunch of benchmarks here. They talk about their schemas and how they approach these different tests. So they have the memory and planning game, and they have street learn. Basically, you can see that they have a sample environment, and from that, you know, from both of those, you know, you have like different places that are visited. You get this learned graph, and this learned graph has structure in it. So in different ways, so street learn is a little bit different from memory and planning, and you can see they're optimizing it, the average reward. So this is sort of a reinforcement learning aspect to it, where they're built, you know, they're optimizing for the average reward over a series of training steps. You can see that it converges at a large number of training steps. And so this is just two benchmarks. So these have nothing to do with development, but they're definitely, like, you know, you can see that they both have graphical structure, and actually strong graphical structure. So if your problem is moderate graphical structure, I don't know how it will perform on what you want to do. But basically, that's the way that they do this. And then, you know, they have other things that are very similar, where you're doing like walking in a room. You have, you know, you vary the room size, and you can see that there's differences in the distance to goal. So, and the number of plans, you know, you can have multiple plans, so you can have multiple networks, and, you know, you look at like, depending on the room size, you have more plans, and, you know, you can make a difference between like different schemas or plans, and you could think of that as like, you know, in the embryo, you know, there isn't necessarily a plan, but they have these hypothesized things called developmental programs. And you could look at those in terms of like evaluating different alternative developmental programs. So, there are ways you could kind of make that connection as well.
Yeah,
So,
Bradley, let
yeah.
me make a connection with that paper we just finished.
Okay.
If you buy seeds, you know that seeds are often rated by viability, which may go down with time.
Yeah.
Okay, and for animals that have multiple eggs, you also have that problem of viability. Only a certain fraction of them produce an adult organism. People don't study the ones that don't produce adult.
Yeah.
Okay, so it could be that those represent some sort of deviation that got out of hand, and so it just couldn't get to an adult stage.
Yeah.
Okay, so viability of embryos or seeds may be a very important thing to simulate or study.
Yeah.
Okay, and I think your thing you did with the Bradenburg vehicles shows that just having a random selection, 30% were still viable.
Yeah. Yeah.
Okay, so what happens if there's any kind of internal feedback mechanism towards viability?
Yeah.
Even though the final, on the way, you may not have a viable structure.
Yeah.
Does the embryo have any means of evaluating its own viability?
Yeah.
This is where this stuff by, I wish he'd join us, Mike Levin comes in.
Oh, yeah. Yeah.
He's looking at these global properties of electric fields, but without considering I don't see that, they're very, very interesting work, but I don't see that he's got an overall picture of how these global electric fields affect development as it occurs.
Yeah.
I may be looking at some of the electric fields in an embryo because, or a cell, because I'm trying to image them with infrared light, and I need to check out to see how it bends, how these electric fields might bend light.
Okay, one thing, Susan, you should be aware of, you know the paper by Jaffe, where he showed that the neural plate of chickens has a huge electric field coming out of it?
Oh, okay. Jaffe?
Yes, Lionel Jaffe.
Okay.
Probably 1970s.
I've heard of them.
Yeah, if you can't find it, let me know.
Neural what? Neural plate?
Neural plate. Electric fields coming out of the neural plate of chickens. A big electric field. What's it doing there?
Not microtubules. The mitochondria have aligned.
Maybe. He didn't look at that detail.
Yeah.
Okay.
I saw a lecture about mitochondria, well, and other things, in the physics lectures, and had an image of a cell, and it was just full of mitochondria, like just full of them. So,
Wow. Yeah.
yeah, there'll be some interference there, I'm pretty sure.
I don't know. I haven't seen papers on electric field effects on mitochondria.
Well, no, but they have a high electrical gradient.
The individual mitochondria probably, but what's the effect of a field on the whole mitochondria?
Yeah, and what is the effect of infrared light on the field and the field on the infrared light?
Oh boy. Good luck.
All right. I think that's all I'm going to talk about for this paper. Again, I can make this available. I wanted to go over this to kind of show the sort of the way that we're thinking about neural networks and graphs and their relationship to some of the data that we've been talking about in the earlier part of the meeting. So again, this is just kind of how they do this with, looking at the wiring and optimizing the wiring for the problem that you're trying to solve. And embryos, of course, we don't have these kind of neat goal-oriented states, so it's a little trickier, but it's still very useful. Yeah. All right. Hello, Morgan. I missed you when you came in, but did you want to say anything or?
Just saying hi and yeah, listening in. Be happy that Michael Levin has a reference to the cybernetic embryo paper in his new Darwin's A Gentle Material paper.
Oh, great! Yeah.
Oh.
Yeah. Yeah.
Morgan, can you send me that reference, please?
Oh yeah, please. No problem. No problem.
That's the first intellectual contact with him I know.
Oh.
Okay. Yeah. No, I think he actually. I think he might have a couple papers of yours on that. It's a massive literature review paper. So it's 20 pages with like 300 references.
Yeah, I've been following him for decades, but I can't convince him to combine his work with the differentiation tree work.
Okay.
I think there's a relationship between the two. Jaffe's discovery of the electric field out of the neural plate may be a key to that.
Yeah.
Okay. The electric fields have a global character. Jaffe did some work on fucus eggs. Fucus is a marine alga, and the fucus egg looks like it's perfectly spherical, but as it develops, it becomes sort of like the Earth. It's got poles on it, and it develops what's called a holdfast. And accompanying that is an electric field that goes through the whole embryo. It's a spherical embryo. And it goes right off one pole, it comes around, goes to the other one. Sort of looks like the magnetic poles for the Earth. Okay. So again, electric fields here seem to be, this is at the first stage before there's multiple cells. Okay. So and I reviewed the work on electrical effects in embryos around the 1990s. And it was a surprisingly few number of papers, but they extended over a period of a century. Okay. Some work on fish, the relationship between fish embryogenesis and electric fields. Okay, so there are weird effects going on, and I wish we could get Levin to work with us instead of going off on his own tangents.
Well, he's been doing some really interesting work at the intersection.
Oh yes, he's made. I think that's one reason he doesn't cooperate with us. He's making all sorts of interesting discoveries, but I think they have to be tied together with the differentiation literature. Okay, so that's there. I know you can't get Mike to come on here and review his stuff, but maybe pick some select papers of his. Maybe Morgan will do it. I don't know. Give us a review of his review.
Gotcha.
Yeah.
Gotcha. Yeah, if you're up to it, Morgan, we'd love to have it.
Yeah. So we didn't talk about organoids today, but Susan said that she was at APS and that they had organoids featured there at the conference. And you're going to give a review of that at some point, I imagine.
Yeah, I'll review this and I'll tell you about the young student with the waves. And I said, did you want, I put my hand up and said, did you want a real life example of your waves? guy said, "Oh, the top of a salamander embryo developing," and he was immediately interested. I don't know if he'll contact you, but I have his information.
Okay, yeah,
Yeah.
Okay.
great.
Bradley, twist Morgan's arm to do a review of Mike Levin.
Okay, all right.
So Mike Levin has a new paper
It's very
out.
twisted.
I maybe need to have that reference as well. So
Yeah,
just I don't send it out there and
all right.
yeah.
All right. Well, yeah, have a good week, everyone.
Okay, take
Thanks
care.
for meeting.
Bye.
Take care. Bye.
Bye.
All right, now I'd like to cover a few papers on developmental biology and biological physics. So the first paper is this paper called Digits in a Dish. And this is an in vitro system. So this is a methods paper published in Frontiers in Cell and Developmental Biology. This is an in vitro system to assess the molecular genetics of hand-foot development at single cell resolution. So the digits are like the digits on your hand or the digits on your feet, which are your toes, fingers and toes. That's what they're trying to do here. So this is largely, I think, for medical applications, but it's interesting because we're modeling things within the embryo, specifically the formation of digits, and so they want to understand the molecular aspects of this. So to gain access to the cells mediating digit and joint development. So they're not just interested in the digits, but also the joints on the digits. We identified a unique property of undifferentiated mesenchyme isolated from the distal early autopod to autonomously reassemble, forming multiple autopod structures, including digits, interdigital tissues, joints, muscles, and tendons. So you have these forming digits, and I think we've talked about in an earlier meeting that you have you have programmed cell death that occurs that reveals digits from it's basically like a paddle or a pad, like a 4-limb pad, and then you get programmed cell death and you get digits that form from that. This is a little bit after that where you start to get the structures that are within the digit. So interdigital tissues, joints, muscles, and tendons. So that's the morphology. Then they're interested in the single cell transcriptomics because that's kind of the thing that everyone's interested in with respect to development are these coordinated gene expression patterns that emerge and that form, are responsible for some of these patterns and some of these structures. So these reveal distinct cell clusters that express canonical markers of distal limb development, including COL2A1, COL10A1, and SP7. This is for phalanx formation. For perichondrium, you have THBS2 and COL1A1. For the joint interzone, you have GDF5, WNT5A, and June. And then interdigital tissues are ALDH1A2 and MSX1. And then for muscle progenitors, you have MyoD1. And then for perichondrium articular cartilage, you have PRG4. And then for SCX, TNMD, which are the genes for tendons. So you can see that they've defined all these different tissue types or precursor tissue types. And they've associated cells expressing these genes that will go on to form these different tissues. So, for example, if you have tissue or if you have cells that are going to form muscle progenitors or muscle, you have those progenitor cells expressing high levels of MyoD1. Similarly, if the cells that you have, which are at this point stem cells or very early precursor cells, if they go down a cartilaginous fate pathway, then they tend to express high amounts of PRG4. So you see that you have this muscle pad. Let me go to our Jamboard here. I'll draw this out. Basically, you start with this paddle, or this, it's almost like a fin, because that's its origins in vertebrates. And then this fin has, all these are stem cells within this. Then you have programmed cell death in between, what will become the digits. So you program cell death in these regions. And programmed cell death through apoptosis is just simply where the cells die with, a certain number of divisions. So that, this is something that's encoded in the genotype so that, the cells can be removed in a timely manner. Sometimes people get webbed fingers and webbed toes if this process doesn't work properly. But once you get this process completed, then you get something that resembles a limb with digits. And so then the digits have to differentiate within their own sort of each digit. So you have cells, of course, that make up the digit. And these cells are going to differentiate into different tissue types, different structures, and so forth. So these are the cells we're talking about. And so, maybe at the distal end of the digits, they might express certain genes. I don't want to be just too descriptive about it here. The joints, of course, are expressing different genes and so forth. So we can identify them at this stage. And then they'll go on to form those tissues later. So this system actually is good for you have to, first of all, characterize the genes that are being expressed in these cells. And then the cells will go on to form these tissues. Analysis of the gene expression patterns for the signature genes indicates that developmental timing and tissue-specific localization were also recapitulated in a manner similar to the initiation and maturation of the developing murine autopod. Finally, the in vitro digit system also recapitulates congenital malformations associated with genetic mutations, as in vitro cultures of Hough A13, mesenchyme-produced defects present in Hough A13 mutant autopods, including digit fusions, reduced phalangeal segment numbers, and poor mesenchymal condensation. So as we talked about earlier, you can have those kind of defects where you have webbed fingers, and this is due to a mutation in one of the Hough genes and the Hough A cluster. And so you'll see this, you can see this recapitulated in their system. These findings demonstrate the robustness of the in vitro digit system to recapitulate digit and joint development. As an in vitro model of murine digit and joint development, in vitro meaning it's in a dish or it's not in vivo. So basically it's cell culture. This innovative system will provide access to the developing limb tissues, facilitating studies to discern how digit and articular joint formation is initiated, and how undifferentiated mesenchyme is patterned to establish individual digit morphologies. So we have mesenchymal stem cells. These little circles that I drew here are mesenchymal stem cells. You're plating these in a dish, so it's not actually on the digit itself. It's in a culture. And then you have these cells here, and they're basically forming digits through expressing these genes and then differentiating properly. So they can actually control this process. Not only that, but they can also produce mutant phenotypes within the culture, and they can evaluate that as well. So this is a nice system for kind of recapitulating development in a dish. The in vitro digit system also provides a platform to rapidly evaluate treatments in that stimulating the repair or regeneration of mammalian digits impacted by congenital malformation, injury, or disease. So this is a way perhaps that we can stimulate some sort of repair or regeneration mechanisms that exist in the digits. We can't do that if we do this in vivo, because we can't necessarily change the fate of cells. We can do this in a dish much more easily, or at least experiment with the different potential mechanisms. The only caveat here would be that in the phenotype in vivo, as opposed to in vitro, you have a lot of signaling that goes on between cells. So a lot of things between cells. There's paracrine signals, for example, where there are other endocrine signals that occur, and that can be different, very different from what you see in a dish. And so this is a nice, at least they've been able to give a proof of concept here. And this might be useful for looking at different congenital disorders and other and just understanding the developmental gene expression of these processes. So digits in a dish, so they were able to get their cells from wild-type embryos at a E11.5 in mice. So they've harvested them, put them in a dish filled with medium, and then plated it, and then they started to play around with it. So this is where you get limb bud dissection and dissociation. So they take like what will become the digits from a developing mouse. They basically take the end of the limb bud, the forelimb bud, they dissociate the cells, then they grow this on tissue culture dishes. They do the single RNA-seq analysis to see what genes are being up or down regulated in each cell. So you get a single cell resolution. So it's actually useful for that. Although you don't have any geometric provenance like you do if you were doing this in vivo. And you also have RNA fish and IHC imaging, so you can image the cells in the dish. You don't have to harvest them and harvest the RNA. So this is actually quite useful for building a proof of concept system that will tell you something about what the cells are doing, but not necessarily in an anatomical context. So you can see that the digits are forming in the dish here. You can see that you have different layers of tissue forming around the digits. It's like a little bit different than what you see in the embryo itself, but basically you end up with digits, and the cells are differentiating in that way. So you see that the formation of digits, not only digits, but carpals, which are knuckles. And so this is all sort of the precursors of this. They'll be from full digits. I don't know how far along the pathway you get of digit development in here, but you definitely get cells differentiating in different ways. So this isn't really an organoid study, but it does, it's kind of a cross between a typical cell differentiation protocol and maybe like an organoid, because I don't think they do this in three-dimensional culture. We see that they call them autopods. Distinct autopod musculoskeletal tissues and structure are present at day 7 in DID culture. So this DID system, by day 7, you start to get some of these tissues and structures that are present in the musculoskeletal system of the digits. And so this is an example of UMAP analysis of gene expression. So this is just showing the sort of the variation of the different cell types and how they segregate out, so we can identify the cell types by different gene expression profiles. That's basically what this is showing. All right, so that's an interesting paper. I think that's useful for like understanding a lot of the coordinated gene expression of the phenotype. This next set of papers is on something called curvatures and condensates. And so these are actually from soft materials, but we're going to now talk about them in the context of development. So the first paper is spatially non-uniform condensates emerge from dynamically arrested phase separation. And so, the formation of biomolecular condensates through phase separation is basically where you get these condensates that are things that precipitate out of liquids. This happens through phase separation. This is the formation of these from proteins and nucleic acids is emerging as a spatial organizational principle used broadly by living cells. So what's going on here is you have these biomolecular condensates that segregate out in terms of spatial organization. So if you're looking at things like different parts of the cytoplasm or different parts of the cell, this is how these things self-organize and self-assemble. So this is an organizational principle used broadly by living cells. Many such condensates are not, however, homogeneous fluids, but possess an internal structure consisting of distinct subcompartments with different compositions. So I think in an earlier meeting we talked about this, where we have these different aspects of like cell condensates. And so if we look at like a cell, we're talking about a minimal cell or some type of model of a cell where we have this is the cell, this is the nucleus, and then this area here is the cytoplasm, and there's some organelles inside this. But the idea here is that all of the cytoplasm here is not a uniform fluid. It's it's a fluid, but it's heterogeneous. And so that term means that you have various things that form in here, and there's spatial organization within the cytoplasm, and that this fluid is heterogeneous. So when we go to model this, we can't model this as a homogeneous substance. We have to model it as a heterogeneous substance with its own dynamics. And I can't remember which paper that was, but we did talk about this to some extent several weeks ago. It was recent. Notably, condensates can contain compartments that are depleted in the biopolymers that make up the condensate. So this is, again, the self-organization of these different biomolecules and how they form rings and how they form clusters. And so we have to understand that, some of these compartments are depleted and some aren't. So if you go back to our model, we have this cell, and there's a heterogeneity to the cytoplasm. If we go to a model with a cell, we maybe just have a nucleus, and then we have several compartments within the cytoplasm, within which maybe different types of metabolic activity occur. And we can model this as like compartments. This is a computational model, so this isn't a real biological model. And the idea is that we would divide the cytoplasm up into these different compartments and simulate them in different ways. So each of these compartments have a sort of set of parameter values that we're going to use. So some of them maybe are thicker fluid, some of them may be a thinner fluid, some of them may be packed with molecules, some of them may have no molecules. So we have to think about, like, what the consequences of our setting up our model are on the future of this material. And so we have to consider during self-organization that things are heterogeneous and not homogeneous, and that different compartments have different properties, and we can set those up in a model and see the outcome. Here we show that such double emulsion condensates emerged via dynamically arrested phase transitions. The combination of a change in composition coupled with a slow response to this change can lead to the nucleation of biopolymer pore droplets within the polymer-rich condensate phase. So the way they're compartmentalizing this through this process of condensate formation is that you have droplets that form. And so the droplets have sort of a sampling of the soup that is this biopolymer within the condensate. It's sort of like what we were talking about with simulating early life and simulating this molecular soup, and vesicles that sample the molecular soup. This is basically the same thing. The droplets are represent basically these vesicles, and they're making the point that just because it's a droplet doesn't mean that it's always the same makeup. The greater soup can be sampled differentially, so that some droplets are very rich in biopolymers, some are very poor in biopolymers. And so this is an interesting point in light of some of the work we've been doing on simulating early life, because it suggests that there's this sort of sampling bias that happens. And this is not something that happens through any sort of conscious intent. It's just a product of the sampling. It's an outcome of the sampling process. Just because it's heterogeneous and we don't know spatially, there isn't necessarily any distinct spatial segregation. But when we draw a sample out of it, either by locking it up in a vesicle or getting droplets out in some way, and in this case it's through condensation, we can get this sort of we can get these differences in number of biopolymers. So they talk about this dynamically arrested phase transition. So we have this change in composition and a slow response to change. So this is a combination that I think we may talk about hopefully more later in the paper. But that's, I think, key maybe to some of this. Our findings demonstrate that condensates with a complex internal architecture can arise from kinetic rather than purely thermodynamic driving forces. And so this is where you have kinetic activity going on, that biopolymers are being made, and that can actually impact this heterogeneity. It's not just thermodynamic driving forces. It's not just energy minimization or just, diffusion processes. I guess diffusion processes would be partially kinetic, but I think in one of the papers we talked about where there were diffusion processes that were, we've talked about this in the past, where you have diffusion processes that are not like a random distribution. They're sort of super normal or super Gaussian in their nature. So that's something to think about. And then provide more generally an avenue to understand and control the internal structure of condensates, both in vitro and in vivo. So they kind of go over what they're interested in here. These spatially structured condensates, they arise in minimal mixtures, and the kind of biomolecules are interested are these peptide RNA condensates. So you can get vacuole formation within them. Vacuoles are just like different compartments within a condensate, but also could be within a cell. They have this core shell structure. You can see this in microgels, for example. In living cells, you see vacuoles in a lot of places, and they can be filled with different types of biomolecules. Nuclear and cytoplasmic germ granules with hollow centers, and poly-RNA nuclear condensates with low density space. Those have all been observed in nature. So I talked about the modeling aspect of it, but in nature you see a lot of examples of this as well. So one of the results that they have is a doubly emulsion structure can form reversibly. So they want to go beyond the formation of this internal structure with a model system containing single-stranded poly-RNA and PEG. So these are co-condensates. So they form in the same condensate. They don't significantly solidify over time and change composition with temperature. We vary the temperature to determine the effect of charging the condensate composition on the formation trap droplets. Liquids pour in these biomolecules inside the condensate. We observe these droplets in condensates at 20 degrees C, but not at 55 degrees C. So there's an effect of temperature. There's also an effect of what they call annealing, which is where you heat the condensate and then you cool it, and you do this multiple times. And then this is something they observe this effect in annealed condensates as well. We conclude that trap droplet formation is reversible and takes place during a composition change. Droplet formation could be a kinetic process or a thermodynamic process. If the formation of enclosed droplets is a kinetic process, we would expect droplets to form at fast composition changes, whereas droplets would also form at slow composition changes if it is a thermodynamic process. So this is kind of an interesting thing. I guess composition changes means what the fraction of each biomolecule is, what the number of biomolecules is per droplet size, and so forth. So I want to get to the formation of enclosed droplets is a kinetic process. A condensate with a radius of 24 microns will contain only one trap droplet after cooling slowly to 1 degree C per minute, but 24 droplets after cooling faster at 20 degrees C per minute. And so basically you get differences in the number of droplets per the amount of cooling. Thus, depending on the rate of composition change, we obtain a kinetic product. a condensate with many trapped droplets inside, a more thermodynamically stable product, a condensate with less enclosed droplets. This observation implies that these droplets are formed when the condensate is unable to reach the thermodynamic equilibrium, in which case we would expect the number of trapped droplets also depends on the size of the condensates. So the size of the condensates and the rate of change in temperature aren't necessarily equivalent. So there's no trend between those two things. Smaller condensates might equilibrate faster with the surrounding dilute phase as opposed to large condensates. So for condensates with a radius of 14 microns, they don't form trapped droplets at any of the tested cooling rates, while condensates at a radius of 38 microns contain more droplets than the condensate with a 24 micron radius, confirming the formation of enclosed droplets is a kinetic process. Okay, I wanted to get to the phase transition part, but I think it's really about thinking about some of these things with, like, droplet formation and kinetics versus thermodynamics. So this is the dynamically arrested phase transition. Combining our observations allows us to understand the mechanism behind the formation of this double emulsion condensate. Consider the binodal of a phase diagram for the poly-RA PEG system, which shows the equilibrium concentrations in the dilute and the dense phase. This is figure 5b, so this is way down. When a change in the environmental conditions occur, the composition of the dilute and dense phase will change to their new equilibrium positions on the binodal. We have observed in multiple systems that if composition change occurs relatively quickly, droplets of dilute phase are formed in the condensate. We conclude that during the composition change, as the system moves from its original to its new equilibrium position, and you'll see with the black dots, the system deviates from the binodal during this change, black lines. So if we go down to 5, we can see this is the phase transition. So you have this initial condition, you have a slow composition change, so it's just changing its composition slightly, versus a fast composition change where you have changes in size and in nucleation. So, and then you have these two different types of change, and then you have this reversing composition change, where it reverses to the original form. So for slow composition changes, it takes less change, less energy perhaps. Fast composition changes take longer and involve more. So here we see this temperature, which is in centigrade. You go up in terms of temperature, and then you go down, and you have these different phase transitions between spinoidal and binodal and spinodal change. So you have this phase transition up here towards the peak temperature, and then as you cool, you get these different phase transitions, or these different states of composition change. And so looking at the diameter of the droplet in microns versus the cooling rate, you see that you get no droplets formed in these dark green circles, droplets formed in the transparent circles, and you see that there's a decrease in the diameter with an increase in the cooling rate, but that the larger diameter droplets always tend, that's the formation of droplets. So this region here is where you get droplets formed within the condensate. Here you get no droplets formed, so the cooling rate can be very large. For a small diameter droplets, you don't get any sort of droplet formation. And so the same thing here, we get this condensate that's maybe at a low cooling or low change in cooling rate, or a low cooling rate. And if the diameter is big enough, you end up with these droplets forming. So this is an interesting finding. It describes this phase transition in some detail. So that's all I'm going to talk about for that paper. For the next paper, this actually goes right to the embryo instead of into these condensate systems. I think the condensate systems are interesting, but they don't really relate to development directly. So this one, however, does. This is curvature gradient drives polarized tissue flow in the Drosophila embryo. So this paper talks about tissue flow during morphogenesis, and it's commonly driven by local constriction of cell cortices, which is caused by the activation of actomyosin contractility. So what that means is that you have a group of cells where you get constriction. And so in constriction, you have where the cells kind of constrict and they introduce forces. And so you have cells like, say, this region, kind of a band, and then they get constricted within like a smaller area, and you get a flow here that opens up. And so you see how changes in the cell volume, changes in the cell position, and there's actomyosin in these cells in the membrane and in the cytoplasm, and they squeeze the cells together to allow this flow. So this is local constriction. It doesn't happen across the embryo, it's just locally. This can lead to long-range flows due to tissue viscosity. So as your tissue gets deformed, there's this viscosity, and it can result in long-range flows. The tissues are all connected by forces, so you can actually get this effect kind of as a ripple through the embryo, and that introduces long-range flows. However, in the absence of cell-intrinsic polarized forces or polarity in forces external to the tissue, these flows must be symmetric and centered around the region of contraction. Polarized tissue flows have been previously demonstrated to arise from the coupling of such contractile flows to points of increased friction or adhesion to external structures. However, we showed with experiments and modeling at the onset of polarized tissue flow in early Drosophila morphogenesis. Drosophila are fruit flies, and we'll maybe see examples of the embryos and how this works. Occurs independent of adhesion and is instead driven by a geometric coupling of apical actomyosin contractility to tissue curvature. So this is related to curvature in the embryo and how the tissues start to curve, and then the outcome is this polarized tissue flow. And this, so yeah, we get these tissue flows in this way. Particularly, the onset of polarized flow is driven by a mismatch between the position of apical myosin activation and the position of peak curvature at the posterior pole of the embryo. So this demonstrates how genetic and geometric information inherited from the mother interact to create polarized flows during embryo morphogenesis. So this is a mechanism that has a maternal inheritance in the egg, and you can see that there's this modeling and experimentation to show that previously proposed mechanisms don't account for some of these tissue flows. So, tissue scale effects can arise as a result of local changes to these cellular attributes in the embryo. So there are attributes such as cell-cell adhesion, osmotic pressure, elasticity, and viscosity. And for example, tissue evagination, which is the folding in of tissues, emerges from apical constriction of well-defined groups of cells. So if we go back to our diagram, this is just a pinching that I showed, but evagination is where things fold inward. And so there's this folding inward over developmental time. The cells fold inward, maybe they form a gut, or they form some other structure like an egg sac, and this tissue flow kind of flows inward so it can make that shape. And so that's one example of tissue flow. This emerges from apical constriction of well-defined groups of cells, such as the mesoderm or endoderm and C. elegans, Drosophila, sea urchin, or ascidians, which are sea squirts. Similarly, apical constriction has been shown to drive cell shape changes during vertebrate development. So when they say apical constriction, they mean there's a constriction at one end of the cell population, and it's polarized. The cells are polarized, so there's this collective behavior going on. Alternatively, anisotropic forces driving cell division can give rise to local divergent flow due to bipolar cell displacement or tissue extension. These two types of symmetric flows may coexist, which is the process of convergent extension. So, during early Drosophila morphogenesis, a single layer of epithelial cells is formed by simultaneous cellularization of 6,000 nuclei, composing the syncytial blastoderm. So there's this large sort of single tissue that forms. It's a single layer of epithelial cells that results when this sort of layer, this single tissue gets cellularized by the formation of many nuclei in a single syncytium. So you get the syncytium, which is a big structure with a lot of nuclei. The nuclei then cellularize, and you get these epithelial cells that come out. And so then this static tissue begins to flow later and during gastrulation and initiates a process of axis elongation along the anteroposterior axis. So we get this pattern of sort of cellularization and then this need for tissue flow to form some of the structure that will come later. We don't really know what creates this polarized flow. It could be due to other processes that are unknown, or other processes that are known, such as mesoderm invagination or germ band extension, but it's kind of a mystery at this point. So this is an example of the Drosophila embryo. This is the membrane, this is the interior of the syncytium, and then this whole area gets cellularized and ends up forming a tissue. So this is the sort of what happens here. You have the posterior end, the anterior end. This is the eggshell on the outside. The dorsal side is on top, the ventral side is on the bottom. What you see is that there's this movement that results in this egg sac at the dorsal side, and then you get so the whole embryo has to move in concert with this to form this cavity. And so you can see that then the cephalic furrow, of course, forms as well, and this requires global coordination. So it requires these long-range flows from local interactions. And so this is the quantification of tissue flow. These cartoons show kind of what's going on, and then these graphs show by position these movements. So you see that there's that the tissue flow moves over time. There's this distinction between the symmetric stage and a polarized stage. So this is when this whole thing is symmetric versus when it's polarized by these different structures, especially the cephalic furrow, which moves down the cell or down the embryo and differentiates things into cells. Okay, so they've performed these same quantifications on mutant embryos for different types of defined mutant, which have blocked mesoderm invagination, germ band extension, and cephalic furrow formation. We still saw this transition to polarized flow. The timing of the transition was even slightly earlier in the mutant ETS embryos than in wild type, which is the non-mutant type, likely due to the lack of ventral pulling from the mesoderm invagination, as evidenced by the increased flow in the ventral anterior region of the embryo. This clearly demonstrates that the polarized flow does not depend on meiosine polarization in the germ band, which is this egg sac that I showed you, or on geometric constraints imposed by the cephalic furrow, as previously predicted. This leads us to two main questions. What physical mechanisms drive tissue flow in the early Drosophila embryo? And what mechanism is involved in the transition from symmetric to polarized flow? So these are two things that they're kind of testing in this paper. They use a mutant to test one hypothesis that removes the dorsal ventral specification of the embryo and leads to the entire embryo acting as a ventrolateral tissue. So just kind of like having a ventral polarization and lateral polarization, but no dorsal ventral specification. So this is a ventrolateral tissue. This makes the embryo rotationally symmetric about its anterior-posterior axis. So in other words, it can rotate around that axis. You have a ventral side, but it changes. It can change as things happen. So basically, the embryo isn't locked into these polarized coordinates. It's free to move any way it likes, as long as it's rolling around the anterior posterior axis. So in tool VL embryos, the posterior tissue does not always flow dorsally, but instead is significant more likely than wild type to flow towards the ventral or lateral directions. So that's interesting. You can get mutants to do different, have different things emerge through these flows. To quantify the flow in the imaging plane, we analyzed only embryos that flow dorsally. Tracking the pulse cells in these mutants revealed that the early symmetric flow is almost completely halted, confirming that the symmetric flow observed is driven by the increased activation of basal myosin on the dorsal side of the embryo. This coincides with the previous work from Strahan et al., which showed that the twist mutant embryos, which have a strong reduction in DV asymmetry or dorsal ventral asymmetry, show a strong reduction of tissue movement towards the dorsal side of the embryo during early stages of morphogenesis. So they're able to characterize some of these movements, some of these tissue flows, and match them up with some of the odd results that people have been getting in the field. So this is interesting. So we next considered what drives the polarized flow that occurs following the symmetric phases of flow. Around the time where the transition occurs, the levels of basal myosin begin to decrease. There's a localized accumulation of apical myosin in the dorsal posterior. So this is, there are a lot of processes going on, and they're able to quantify the flow by pulse cell tracking. We concluded that the symmetric phase of flow requires a non-uniformity of basal myosin, and the polarized flow requires apical myosin. Since polarized tissue flow is normal in ETS mutants, in which mesoderm invagination and germ band extension are blocked, we conclude that apical myosin is required strictly in the posterior region of the embryo and not in other adjacent tissues. So this kind of goes back to the last paper with condensates and just kind of this self-organization or the self-assembly process where things aren't meant to be homogeneous. They're highly heterogeneous. And sometimes it's a matter of just sampling or like regional difference. So in other words, if you have some process going and you just take a sample from one area or another, you'll get some sort of order that can be amplified later. So this is kind of an interesting effect. I don't really know I'm not going to say any more about the connection between the two papers in that way. But I will say it's an interesting parallel. So this is an example here again of the Drosophila embryo. This kind of shows the velocity in microns per minute of these cells around the edge. And you see that there's this tissue flow here where there's this bending. And you can see that there's movement here. And then they show the basal myosin, this V bar, which is, I'm not quite sure what that is, but V bar minute, microns per minute, I guess that's the velocity. And then the position PC microns, and you can see the results here. Basically, it's increasing over time. In this case, you get this difference between the wild type and the mutant. In this case, the mutant starts higher and kind of gets to the same place. In this case, I think the mutant is this one here. So there are two mutants. They're less active than the wild type in terms of position. And then the basal myosin, there's invariant sort of expression of that in the mutants, but not in the wild type. So you see that there's some differences between the mutants and the wild type. They point out that localized friction or adhesion is not responsible for polarized flow. So this idea of using friction or adhesion as a way to explain it is not exactly what we want as an explanatory framework. During the polarized phase of flow, the dorsal posterior activation of apical myosin leads to bending of the epithelium. This causes the anterior end of the apical myosin domain to come in close contact with the eggshell, which could create a localized domain with higher friction. It could also lead to adhesion between the epithelium and the eggshell. Intuitively, this configuration could lead to asymmetric contraction, but adhesion at the anterior end of the apical myosin domain has even been shown to be crucial to the anterior wave propagation of the endoderm invagination, slightly later in development. So they test this using the polarized flow we observe could be created by such an asymmetric friction, which of course is what they did here, and it did not show that. And so they conclude that interaction of tissue curvature with active moment can explain the polarized flow. So now we're talking about interactions of tissue curvature with the active moment. So polarized forces in this polarized flow can be created by the interaction of a curvature gradient. So that's just the cells as they curve and the interaction of that gradient with an active moment, which is from the theory of active surfaces. And so active moments are created when apical and basal myosin tensions differ. So there's a difference in tension, and that can create what they call an active moment. When myosin, and therefore active tension, is present at similar levels apically and basally in a region of tissue, this acts to contract this region, which in turn exerts forces on the surrounding tissue. So when we go back to this diagram here of the forces and these flows are really the forces acting downstream. So there are things going on here. They're resulting in transfer forces that go down this area of cells. And so you get these long-term effects through that. So when the levels of apical and basal myosin differ in a region of tissue, an active moment is present that causes this region to exert torques on the neighboring tissue, which then in turn acts to increase or decrease the curvature of this region. So they summarize this in this figure. Active tension force here that are pushing together. This acts to decrease the length of the green region, which is this. This is active tension pushing in a bunch of cells. And then the active moment here is where you get a moment, which is where you get this torque, and it's acting on this green area of cells, just like in A, but then instead of pushing it together, it pushes it down, and it decreases curvature on the green region, and that's what we have here. And so again, you can see this in the embryo here at the edge. You get this green region. This would increase curvature of the green region. This would decrease curvature of the green region, depending on the sort of the apical versus basal forces and the interaction there. So hopefully you found that interesting. Hope you learned something.